Hello. First of all, thanks to the organizers for putting together this exciting meeting. My name is Marcin Joachimiak. I'm a staff researcher at Berkeley Lab working with the DOE K-Base. And today I wanted to tell you about our machine learning project to uncover microbial function indicators for Earth biomes. K-Base has been a Department of Energy project since 2010. Recently we moved into a new green building just before the pandemic, so it's still new. And K-Base is a system that aims to empower scientists and teams to collaboratively perform computational biology around the function of plants, microbes, and their communities. More recently, we also have an internal R&D project called the Knowledge Engine to support prediction and inference on the system. Metagenomes are a primary data type of interest for K-Base. However, as you may know, there are some long-standing data and metadata standardization challenges in this domain. Nevertheless, we're beginning to see some large meta-analysis. This is work from the Bork group, where they analyzed 13,000 metagenomes across 14 habitats using cogwhirls and constructed the Similarity dendrogram, where we see that soil and skin environments are similar. This is intriguing to us, and we wanted to see whether this result would withstand the test of time with more data and better classifications. We turn to the EBI Magnify to access standardized metagenome data and metadata. EBI publishes their pipelines on GitHub. They rely on Interpro for annotations, and they're also able to curate some of the metadata, including gold classification biome paths that we use as our primary labeling. Here I'm showing you a t-SNE analysis of 32,000 Magnify samples across 122 biomes, and this is over 4,400 Go terms. So already you can see that this data space is much larger than previously. And however, there's quite a bit of mixing of different environments. You can see that hosts associated and environmental often are placed together. It's a fairly complex, high-dimensional view here, and you can keep this eel or worm image in your mind to make a connection later in the talk. I also need to note that about 3/4 of these samples are from human, so there's a strong anthropomorphic bias, which can be a bit disappointing if you're interested in the environment. Our primary machine learning task was biome classification. As usual in machine learning, most of this work was upstream. We used the Magnify API to pull metagenome abundances and metadata, and then through a series of machine learning best practices, including filtering out correlated features, we arrived at a dataset for training, which included 1,780 Go terms, 32,000 samples, and 122 biomes. For machine learning methods, we tried TabNets and the multi-layer perceptron, so table learning methods. However, what worked well in our hands were graded boosted decision trees as implemented in the CatBoost package. Decision trees have a very nice property where you can explain these models using Shapley values and feature importance, even at the per class level. And moreover, they are able to include absence of features as positive information for the model. So absence of a Go term, as you can imagine, could be important for distinguishing different biomes. We performed model evaluation and selection and also worked with feature importance. To identify robustly an important set of Go terms, we performed feature permutation analysis using multiple CatBoost runs. We computed the average rank above the highest permuted threshold and identified 86 features that we used to build the final model. These are results for that final model. The confusion matrix is on the left. Along the diagonal, you can see the intensity of prediction performance. In most cases, biome prediction was perfect or near perfect. However, there are a few biomes which cause problems, notably brackish water and estuary. We think these are more complex, perhaps mixed environments that may also have some time and space sampling issues, and there's always a question of classification resolution for the environments. Our 50-fold cross-validation results were 0.89 on the test data. We're able to extract the feature importances and analyze them. Here is a table showing Go terms on the y-axis and biomes on the x-axis, and I'm going to show you some feature importance extremes. So for 2 animal fecal microbiomes, we find that alanine transport and modulation by virus of host process are indicative of these environments. And this corresponds to a potentially nutrient-rich environment as well as presence of viruses. We now have some evidence that important functions can help resolve metagenome differences. We still see overlap between host-associated samples and some human samples. We think this is suggesting label improvements, And some direct evidence for this is coming from the model itself, where fecal microbiomes from mammals are most often predicted to be human, as seen in this confusion matrix off diagonal. We can also repeat the t-SNE analysis, now restricted to the 86 important Go terms versus the original 4,400 Go term set. And in this view, we see some better separation of environmental and host-associated samples, occupying their own regions, separated from other high-level biomes. There's still, of course, some mixing happening. In fact, the host-associated samples overlapping with the green color here are again suggestive of potential sample relabeling. And then finally, the orange points, the engineered samples, as I mentioned, are often sampled from the environment, but then worked on in the lab or in industry, and hence we see them sprinkled throughout. Using the important functions in biome data, we can also compute biome similarity networks. We think that networks have some advantages here. In contrast to dendrogram, they're not limited to parent and child relationships, and in contrast to the t-SNE, they do not include all the data and rarefy to the highest and most similar relationships. So we compute pairwise biome correlations and apply a high threshold. And on the left, I'm showing you the result of that, so a 2D layout of a network, colored by high-level biome categories. We see some clustering here of host-associated, engineered, and environmental samples, with engineered samples for the most part being between the other two categories, so perhaps indicating some kind of flow between human and built environments and the natural environment. And then on the right, I'm showing the same network, but now colored by node degree, and with the node size representing centrality. Warmer colors are higher degree. And here the highest degree in most central environments are pond sediment and industrial wastewater, which we believe are environments that interface with multiple other environments, as well as potentially including this human to natural environment link. We can transpose this matrix and can now construct a functional similarity network across the 86 Go terms and the biome data. Again, node size represents centrality, and the node color represents degree. Warmer colors are higher degree. We see a dense subnetwork on the right and a sparser subnetwork on the left, and separating these two is this kind of gateway function, which is amino acid transport. We think this is a delineator between nutrient-rich and nutrient-poor environments. Furthermore, within the dense subnetwork on the right, there is an antibiotic catabolism function, which is the highest degree and most central. And as we know, humans are large consumers of antibiotics and continue to stress our microbiome populations with antibiotic stress. Finally, we see a few viral-related terms, which we know are related to the host-associated samples, so this dense subnetwork on the right represents functional relationships from that space. More results and more interpretations will be in our paper coming up soon. To conclude, I showed you our biome classification results as a useful machine learning task, but along the way we also learned about important features. So now we have an expanded set of use cases. First, we can apply the model to recommend standardized biome labels, and also to suggest sample metadata relabeling. The important functions can help interpretations in the complex landscape of metagenome samples. But most importantly, the reduced function set can lead to efficiencies in future analysis. So some examples of this are searching with a restricted set of HMMs to perform annotations, or restricting the function space to perform analysis. Of course, additional features can always be recruited via Similarity. Finally, I wanted to acknowledge all the people involved in this work. So first of all, the overall DOE K-Base team, and then special thanks to the K-Base Knowledge Engine team, and in particular Ziming Yang and Xiang Yungblith. Please feel free to reach out with any questions or comments, either in person here or online. Thank you.